Hi, this is Julia Wagu,
And this is Cynthia Robio.
and the title of our article was Mechanism of Adaptation. A549 cells adapt to high concentration of nitric oxide through bypass of cell cycle checkpoints. It was by Aqil M. et al.
And we are in Dr. Sadana's class, Cell Biology 4320, and our topic was the cell cycle. As we mentioned before, our topic was the cell cycle. But before discussing our research article, we'll go ahead and give a small introduction and a reminder as to what the cell cycle entails. There are 4 phases in the cell cycle. Starting with G0, G0 is a resting phase, and it's a period in the cell cycle where the cell exists in a resting state. G1, in the G1, the cell grows in size and synthesizes mRNA and various proteins that are required for DNA synthesis. The S phase is the part of the cell cycle in which DNA is replicated, occurring between the G1 and G2 phases. Precise and accurate DNA replication is necessary to prevent genetic abnormalities, which can often lead to cell death or disease. The G2 phase is the third and final subphase of interphase of the cell cycle, and it directly precedes mitosis. It follows the successful completion of the S phase, during which the cell's DNA is replicated. The M phase, or mitosis, is much shorter than interphase and lasts only a couple of hours, and it contains a metaphase checkpoint that ensures that cells are ready to complete the cell division. The cell cycle consists of various checkpoints that serve as halting points prior to proceeding to the next phase. Those are the G1 to S and G2 to M, as well as the metaphase checkpoint that ensures that the cells are ready to complete the next cell division.
These checkpoints consist of various proteins that help them go from one to the next. The first protein we're going to talk about are the cyclins. These are activator proteins in the cell cycle. They connect with CDKs, which are also known as cyclin-dependent kinases. These protein kinases exhibit enzymatic activity only when they are bound to a cyclin. The time a cell may spend in the cell cycle varies from cell to cell. So, Cynthia, can you let us know why the cell cycle needs regulation?
Of course, Jewel. So the cell needs regulation at each phase in order to proceed with caution, much like traffic signals. They give us the OK go or hold on to your horses. The cell does the same, as it may need some extra time in one phase, making sure that the cell is fit to carry on in the cycle.
So next up, we're going to go over some terminology pertinent to our article that may ease our discussion. The cells used in this study came from A549 cells, which were deemed the parent cells. These cells served as the control group.
These were then compared and tested against the A549 HNO cells, which were adapted tumor cells that contain high amounts of nitric oxide.
They both received nitric oxide donor treatment. Nitric oxide that is induced into cells acts as a perfusion enhancer, increasing blood flow. This is related to cancer cells' ability to initiate angiogenesis, which is the development of new blood cells.
Our article also mentions the P53, which is a tumor suppressor protein that can initiate apoptosis.
Another protein utilized in this study is P21CIP1, which is a kinase inhibitor protein, which is anti-apoptosis.
As we mentioned before, the cell uses various components to regulate itself, one of which is the CDC25A. It regulates the cell cycle through dephosphorylation and thereby activating cyclin-dependent kinases. It functions continuously throughout the cell cycle. It activates both CDK2 cyclin E and CDK1 cyclin B.
The image on the right shows the A549 cell used, and this image was captured using phase contrast microscopy.
So now we'll go ahead and take a look at the A549 cell versus the A549 cell HNO. In order to observe the changes in the cell, both cells, like Jewel mentioned earlier, were induced with nitric oxide, abbreviated NO.
The study analyzed the effect of nitric oxide on various components of the cell cycle, such as the CDKs, which are also known as cyclin-dependent kinases, cyclins, which are the activator proteins, and other helper proteins.
Several time frames were recorded and evaluated utilizing line graphs, densitometry analyses, and Western blotting techniques.
The figure on the left depicts cyclin D3 expression in a treated A549 and A549 HNO cell during 0, 8, 16, and 72 hours respectively. Okay, now we're going to go over the adaptations observed by nitric oxide treatment in CDKs and cyclins. So in the cell cycle, the CDK4, CDK6, cyclin D complex is associated with the G1 phase. During late G1, the CDK2 cyclin E complex is activated, which facilitates entry into the S phase. The CDK2 cyclin A complex is important for maintenance of the S phase, whereas CDK1 cyclin A is important for G2. Now, for entry into the M phase, the CDK1 cyclin B complex is activated, which is critical for the G2M checkpoint and maintenance of the M phase.
The HNO cells are able to proliferate under nitric oxide exposure, which prompted this investigation to compare the expression of patterns in the CDKs and cyclins between the parental and the HNO cells.
Figures 4 and 5 are densitometry and Western blot analyses of various cyclins and CDK proteins in A549, which is the parent, versus the A549 HNO cells at early and late time points after nitric oxide treatment.
As Jewel discussed figures 4 and 5, we're going to go ahead and look at each graph and Western blot specifically to go ahead and go into greater detail at every phase. So if we look at the G1 phase, we have CDK4, and it shows that the expression was strongly induced by the nitric oxide donor in parent cells, and it approached similar levels to those of the HNO cells. In HNO cells, it remained high at all time points, and it did not show much change with the nitric oxide donor treatment. In contrast, the expression of the CDK6 decreased initially in both the parental and the HNO cells following the nitric oxide exposure. However, it increased in HNO cells in a nitric oxide dependent manner at 16 hours. With prolonged nitric oxide treatment, the levels in NHO cells decreased compared to those in the parent cells. Cyclin D3 was upregulated in response to nitric oxide in parent cells. In the HNO cells, it remained high at all time points, independent of whether or not it had the nitric oxide donor. With prolonged treatment for 72 hours, cyclin D was high in the HNO cells, just as CDK4 levels were similar at the 72-hour mark. The nitric oxide responsive expression patterns of CDK6 suggest that CDK6 cyclin D3 may specifically operate in G1 phase under nitric oxide stress in A549 cells.
Now let's take a closer look at the G1S transition as well as the early S phase. The study checked for expression of the cyclin E CDK2 complex. CDK2 was upregulated initially in response to nitric oxide treatment in parent cells till 8 hours, but started decreasing at 16 hours. You'll see in HNO cells the CDK2 expression was independent of the nitric oxide exposure. With prolonged exposure, the levels were in the same place for both cell types. Cyclin E levels also increased in both parent and HNO cells in response to nitric oxide exposure. However, such a high level of expression was only kept in the HNO cells at 16 hours as well as 72 hours. The high level of cyclin E CDK2 complex suggests that the G1S checkpoints were impaired in the HNO cells. This can lead to an accelerated entry of HNO cells in the S phase.
Now, as we talk about the S phase, for maintenance of the S phase, the cyclin A CDK2 complex is important. Thus, we assess cyclin A expression status. Cyclin A levels increased initially in response to NO treatment in both parent as well as HNO cells. However, with prolonged exposure at 72 hours, the cyclin levels were similar in both cell types. Now, during the late S phase, cyclin A CDK1 complexes are formed, and they are important for the G2 phase, the next phase. whereas cyclin B and CDK1 complexes are crucial for the G2M checkpoint.
Now we're going to go over the adaptations observed in P21CIPI. Now with prolonged nitric oxide donor treatment, the P21CIP1 levels increased in HNO cells to very high levels. This could result in the inhibition of the cell cycle at G1, G2, or S phase, depending on which cyclin CDK complex it is binding and inactivating. However, it has been reported that the cyclin D3 CDK6 complex is not inactivated by P21CIP1. Cells utilizing this complex in G1 phase can still bypass high levels of P21 CIP1 and enter S phase. High levels of P21 CIP1 can lead to both pro-apoptotic and anti-apoptotic signaling depending upon the cellular context. It can bind to apoptotic signaling kinase in the cytoplasm and inhibit the MAP kinase pathway. It can also bind to pro-caspase 3 and prevent its processing and activation.
It is important to note that high levels of P21 CIP1 protect cells from P53-dependent apoptosis during DNA damage and induce cell cycle arrest. Therefore, inhibition of apoptosis and restriction of HNO cells in S phase might be due to the same levels of this P21 CIP1.
The levels of the cell cycle inhibitor proteins P21CIP1, P27KIP1, and P15INK4B were also assessed. P21CIP1 was drastically upregulated in parental cells at the 8-hour time point compared to the 0-hour after nitric oxide treatment, suggesting the activation of the G1 checkpoint in parental cells. It was high in HNO cells initially and gradually reduced with NO donor exposure. It was completely diminished under NO exposure at the 16-hour time point in both parent and HNO cells. At prolonged exposure at 72 hours, the HNO cells showed much higher levels of P21, CIP1 compared to parent cells. This is consistent with the cells attaining quiescence and staying in G1 phase after long exposure of nitric oxide. The expression level of P27K1P1 was reduced with extended nitric oxide treatment in both parental and HNL cells, although at a moderate level. The expression of the level of P15INK4B was also moderately reduced both in parental and HNO cells in response to nitric oxide treatment.
Expression of P21CIP1 is transcriptionally regulated by P53. Sustained and enhanced P53-P21CIP1 DNA damage checkpoint activation in A549 HNO cells may also correlate with enhanced DNA repair capacity to maintain the genome integrity of A549 HNO cells. If we take a look at the figures 6 and 7, we have the densitometry and Western blot analysis of the cell cycle inhibitory proteins of both A549, the parent, as well as A549 HNO cells, both at early and late time points after the nitric oxide treatment. It's evident that at the 72-hour only, the P21CIP1 expression status was determined.
It's also important to note that beta-actin was used as a mode of comparison.
Now let's go ahead and wrap this up. This article found that checkpoints are an integral part of the cell cycle as they contribute to the importance of DNA damage recognition and cell cycle regulation in providing resistance to tumor cells that are HNO expressors.
It is well established that deregulation of the cell cycle is a hallmark of cancer development and progression.
During long-term nitric oxide adaptation, A549 HNO cells gradually acquired and adapted an enhanced cell cycle regulation network, which distinguishes the HNO cells from parental cells in response to the nitric oxide exposure, and it also leads to improved survival in harsh conditions.
P21CIP1 can be used as a potential biomarker to profile patients with resistant tumors to be able to provide the best possible drug combinations. This concludes our presentation. Once again, I'm Julia Nwagu.
And I'm Cynthia Robio, and I hope you enjoyed our presentation over a mechanism of adaptation. A549 cells adapt to high concentration of nitric oxide through bypass of cell cycle checkpoints by Aqil M. et al.
Thank you.